We discovered the use of a drug that kills human cancer stem cells, but seems to have no effect on normal stem cells. The unusual aspect is the way it actually kills these cancer stem cells is it differentiates them. It changes them into cells that are non-cancerous instead of working the way normal chemotherapy works, which is to be toxic to the cells. We think this lack of toxicity is why it doesn't have an effect on normal stem cells, which we think is going to be beneficial for the patient. We discovered the drug by creating a new way of looking at different chemicals that would kill cancer stem cells but do nothing to normal stem cells. Now, to be able to do that, you have to be able to put cancer stem cells, human versions, in a dish, but also have normal stem cells to also test with the compounds. We were able to do this with a robotic system, fully automated, that allowed us to go through 10 or 15 compounds. Now we can do this with thousands of compounds, eventually arriving at this drug that doesn't do anything to normal stem cells but kills cancer stem cells. We are very excited about this drug, and our next steps are to try to use this in clinical trials. Our focus population will be adults with acute myeloid leukemia. These would be patients that unfortunately have relapsed from the disease using conventional, currently available chemotherapy. So we want to test this drug and to see whether we can get these patients into remission and reduce the leukemia burden. What's most interesting to us is that this drug actually binds a receptor which is on the surface of the cell, and we've also identified that this receptor is found on cancer stem cells of poor prognosis of breast cancer. This is important because we might be able to use this as a biomarker, basically being able to detect the cancer very early and they'll be able to treat it earlier in breast cancer. So the fact we've gone from leukemia to breast, we're also hoping that this same drug might be effective in other types of cancers that we haven't looked at, and that's certainly our next set of steps with this drug. Our platform, however, has identified another 12 candidate drugs, all of which need further characterization, but they worked very similarly to the drug we've discovered here, and we're hoping that they will also have effects on multiple types of cancer. Well, certainly we want to characterize the drugs we've already identified, but there's no question that our capacity now to do this with thousands of compounds means that we should probably be looking at lots of other chemical libraries that people have all over the world, and we've started to develop those collaborations right now with many academic groups. But I think we need to industrialize the process in a way that partnerships with the private sector will be critical moving forward. The goal is the same, however, which is to find really unique and novel drugs to change the way we tackle and treat cancer.